Okay, yeah, I'm going to talk about the small scale production of biochar based on 5 materials and then the analysis of these materials. Um, this is part of my PhD here in Dublin, and it's like the results I found within the first 2 years. So it's just the basic analysis I've done so far, as the whole purpose of the PhD is more to see if the biochars I actually produce will be able in the end to absorb greenhouse gas emissions from cattle manure. I will be testing here in Ireland. Um, so yeah, as Jorge already said, there are like various types of pyrolysis reactors out there, and he used the rotating one, whereas due to limited applications here in the lab I focus also on the muffle furnace, also to see if the pyrolysis reaction actually works on a very, very small scale as well. So what I did is, as you can see on the picture on the bottom right, that's the muffle furnace I used. And for that, I ordered a few crucibles, ceramic crucibles, to which I then added the feedstock, and which were then closed with a lid before being inserted into the furnace. And the furnace was just closed as it is. I didn't have any nitrogen flow or anything, so it was really just a basic production everywhere, like you can do in very small scale, very simple setup. And I produced the chars from either the fresh material as I got it, or also from dried material, just to see for the production if there is a difference in the yields, or if, like for example, some characteristic features actually vary. So the drying of all the feedstocks and the dry matter determination of the materials for that matter was done at 105 degrees for 24 hours, as it's like a standard method out there. And then I focused in the beginning on pH analysis and dry matter content. And further analysis I've done recently was SEM-TGA and some spectroscopy and surface area analysis as well. The biochars were produced at 4 different temperatures, and I got those from the literature, as there seemed to be the most common one, just to be able to in the end compare my analysis with those as well. I chose a residence time of only 20 minutes to make it still economically viable, because like if you keep it for a long time, you automatically need more energy to produce the biochars, which maybe on a large scale, especially on or on a farm scale, which might work later, it might not be the most appealing for actually the consumers. And I used a heating rate of 7.5 degrees per minute. So the feedstocks I used were supplied by me from a company here in Ireland, and we went to pick them up down to a bit south of Dublin to Kilberry, and the materials we found there was just surface peat, which didn't have any volume at any value at the time to be used to be turned into horticultural peat, or like to make briquettes or anything. So the company was looking for a way to make use out of this waste material, which they couldn't add any other value to. Another feedstock I'm using is the peat fibre extracted from that peat, which is too big of chunks to actually be inserted in the normal horticultural peat as well. And then we got fine urban green waste, which is a bit pre-composted mixed green waste collected from the region there in Ireland, and then it's just splitted depending by the fraction, and the fine ones are like the smaller ones. You can, it looks a bit like normal black, like ground or soil, just to give an image. The brewery grain is spent grain, which is the residue they got from a brewery company here in Ireland, and which the company didn't have any use for, and then they were selling it to the company, to the waste company, to actually get rid of it, and the company thought maybe they can add some more value to a product they keep getting continuously as well. And the pictures on the right side of each name is basically just an example of how the biochars looked after being produced, just to give an example of how the feedstocks were treated, or that they actually didn't really change the shape even due to pyrolysis and being inserted into the crucibles for the process as well. So at first I focused on the analysis of the feedstocks, just to have backup parameters to actually know with which kind of materials I'm dealing with. And it can be seen that the brewery grains have the highest dry matter content, as they were already dried at the facility after the beer was produced, whereas the fine urban green waste had the lowest dry matter content, as seen on the pictures before. It is just lying openly on the facility in piles, and it's subject to every condition, and everybody knows Ireland is not really a country where you get a lot of sunshine, where it's more like you get rain a lot, which means all feedstocks lying outside will be subject to a lot of humidity as well. And then I got some lignin content results showing that the peat and the peat fibre actually contain the most lignin, which makes sense if you know how peat is actually developed throughout hundreds of years. So it's basically just raw, like wood, not raw, but like processed wood or things buried in the ground for a long time and just breaking down very, very slowly, which explains the result here as well. A bit surprising was the result for the brewery grain, for the white one, although it might have been that in that particular analysis, in that particular batch, they more had the powdery than the actual spelled ones, which might be able to explain those results for that feedstock. As a next step, I produced the biochars first, as I told before, from the fresh material and then from the pre-dried one as well, just to be actually able, if you calculate it on the dry matter content of all the feedstocks, to actually say if the yield would be the same. So if you have a feedstock all continuously available and you can use it up in a big scale, if it actually makes sense to dry it and therefore increase the cost of production, or if it actually makes sense just to use it as you get along. And I know the results look very much like that. The dry matter yield is, or the yield produced by the dried feedstocks is way higher. But if, for example, if you keep in mind that the brewery black just has a dry matter content of 90%, and then you can, if you compare then the two results there, you see that the yields are actually quite along, so they are comparable, which I followed from that you actually do not need to dry the feedstocks if you use them up as they come. If, for example, if you need to store them for a long time, of course it would make sense to dry them before, because otherwise they would be breaking down over time. And in general, it could be seen that, which is probably known by many of us already as well, is that the higher production temperatures you have, the lower yields you get. And this was seen throughout all the 5 feedstocks I used in the project as well. For the pH values, I obtained those with a method described in Biochar analytical guiding method. Sorry, I missed the title right now. But it's basically you're using a 1 to 10 weight to volume ratio for feedstock and deionized water. Shake the obtained liquid for 1 hour, let it settling down for 30 minutes before you're actually obtaining the pH values. And what can be seen here is that while most of the feedstocks had quite an acidic pH, higher treatment temperature actually led to pH values in a basic area and making them increasingly higher with from 450 to 750 as well. And it can be seen as well that, for example, both feedstocks have a slightly lower pH in comparison, for example, to the brewery grain seen here as well. Then I was able to obtain a few TGA results over in Birmingham together at Porter's laboratory as well. And the results I have so far obtained from there are just the moisture content and the volatile matter content, which you can see here. And it can be seen that whereas the volatile matter content is continuously decreasing throughout treatment temperatures, the same cannot be said for the moisture content. Though a few backup trials will be done to actually confirm those results, as I haven't seen those in moisture in dry matter analysis I've done over here just in a normal oven. In general, it can be said that the moisture content varies between just 3 and 5% of the total volume, and the volatile matter content varies between 10 to 38% on a calculated on a dry-based material. As a next step, I was able to determine the surface area of those biochars, and all those results I've shown you previously are just based on the dried materials. So everything I'm presenting here is not focused anymore on the wet materials, but also only on the pre-dried material to make it more able to compare to the literature results as well. It can be seen here that the peat samples actually do have the highest surface area, whereas the brewery grain and the green waste actually stay more have lower surface area, just ranging around 100 cubic meter per gram. I am sorry, there seems to be some problem. I don't know what is happening. And I'm sorry that I'm not answering right now the questions from the chat, but it is a bit confusing to focus on both. Um, I'm sorry. No, that was not supposed to be the next one. Okay. From within the same analysis, of course, I obtained not just the surface area, but also the micropore area, as that might be interesting for my later emission trials. And it basically shows the same results of, or similar results already as the surface area ones, that the peat samples have a bigger area, whereas the brewery grain samples have a lower surface and micropore area. And I hope now it keeps working. I'm very sorry for this issue. It worked before perfectly. Okay. Yeah, as a next step, I obtained a few SEM pictures, and this is just like an impression of all the pictures I actually obtained. On the left, you can see the original feedstock and the scale in centimeters to get an image on how big the feedstock is, or how diverse it actually is in its composition. Whereas on the right, you can see examples of the biochars produced at the different temperatures from the pre-dried material. And it can be seen that even at 750 degrees, the samples are still able to contain their original properties. For example, the image I chose here for 750 degrees was a chunk of wood being present in the peat when it was collected, and even after 750 degrees, you can still see the shape hasn't really changed. Whereas it can be seen as well that throughout treatment temperatures, you get a nice porous structure, and which can actually help maybe in the future as well to absorb the gases as well. So the next will just then be the same temperatures, the same temperatures as before, just for different materials on the following slides. For the peat fiber, most of it were basically the chunks, what I showed before for the 750 one, as this is the material which cannot be sold as gardening or growing media. It can be seen that whereas at 450, the material is still pretty dense and just has very few porous structures or holes inside, the higher the treatment temperature you actually get, the more pores you get, and the more surface area you get, as also have been seen before on the diagram as well. And I find it really interesting that the shapes they get more, you can basically say not artistic, but it looks more like paintings, and you get really uneven surface structures, which might for future purposes might be able not only to absorb the gases, but might, if it's then later applied to the soil, might to add some value for that as well. For the fine urban green waste, you could see, as for the peat in the beginning as well, that it actually keeps its structure. It keeps being this small crystal round balls of material which just start growing to be more and more porous depending on the treatment temperature, as seen here in the pictures as well. Whereas the brewery black really, it stays, the pictures I've shown here were specifically to see how one particle of that brewery spelt actually evolves through higher treatment temperatures, and it keeps the outside stays the same, but the inside, the pores get higher, the pores get larger, I'm sorry, but the surface gets more complex as well, and it's not as like flat and anything anymore as it was in the beginning, but it actually starts to interact with all the other material around it, and starts to actually maybe absorb some of the things sticking to it as well. The white one, as seen here on the picture, is mostly what I got is a very powdery material, and therefore even within the powder, the structures really get very complex, and it will be interesting to see for the future how they are interacting when they're applied to the manure. But the limited of residue of spelt you actually have left gets very porous, even though it didn't show in the SEM results. And yeah, so at last I actually was able to obtain a few FTIR results, which are supposed to, which are supposed to show, as seen here, that for the brewery black, the peaks and dents are, even throughout the treatment temperature, are at the same places, out of which I figured that they have the same functional groups on the outside. And the ones obtained in that analysis was that you can see that the biochars produced from that specific material have not only hydroxy or carbonyl groups, but also carbon bonds at the same time as well. So whereas the brewery black was clearly differentiated by the temperature, it can't be seen here for the one biochars produced from the brewery white grains, because they are very overlapping and not as clearly, the aromatic bonds are not as clearly actually readable as it was before. As you can see here, the pattern is much more scattered, has much more noise in it, which I still have to read out and trying to get rid of. But it can be seen that you have similar bonds from biochar white and biochar brewery white and brewery black materials. So to conclude, of course, it can be said that if the materials vary, the properties of the biochars will vary as well, and that the moisture content and the volatile content decreases with higher treatment temperatures, which makes the biochars more stable, from which it could actually favor higher treatment temperatures. Also, another point which might favor higher treatment temperatures is actually a higher surface area as well. And the next step I'm going to pursue is an ICP, so an elemental analysis of the biochars, as well as further surface area analysis to validate the results obtained, and to start my emission trials as well, preferably with the equipment seen on the right side here as well. And therefore, I want to thank the partners who were able to, with whom I was able to undertake all this analysis. And if anyone has any more questions, please, get back to me, and I will be able now to maybe answer those from the chat as well. But yeah, if you have others, please let me know.